Good. All right, good evening, everyone. Good evening. Welcome to 2022 Gilfillan Lecture. Good evening. Yeah, I never really learned a loud version of a whistle, so. Well, welcome, everyone. Thanks so much for joining us for the 2022 F.A. Gilfillan Memorial Lecture. Tonight, we're so privileged to hear from one of our esteemed colleagues, a globally renowned molecular geneticist recognized by the American Association for the Advancement of Sciences for his extraordinary work in fungal genomics. So we're here in person tonight, which is great. It's 3 years ago when we were last together for this lecture. In person. Last year we had an opportunity to do it remotely, but it's nothing like being in person. But we're not alone in person. There's about an equal number of individuals, roughly 100, that are remote with us this evening, and wonders of technology. I've often said in recent months that if we put together a faculty committee to design a future in which we could do remote lectures, we would have started, and we'd be about 5 years away from figuring out how to do that now. Instead, we decided to do it in a weekend. So it's nice. Anyways, it's nice to take a few of the positives out of the pandemic, like the use of Zoom. So colleagues all over the country, colleagues, friends, and family from all over the country. We're joined by as I mentioned, colleagues, alumni, friends from OSU, colleagues and friends from University of Oregon, from Johns Hopkins, from New Mexico State, Washington State, and many other science colleagues nationwide. And a special welcome and thanks to the Gilfillan family who are with us on Zoom, and maybe one or two here in the room. Before we proceed, I want to acknowledge the impact of Oregon State's land grant history as it's had on the indigenous communities in Oregon. The Morrill Act of 1862 cleared the way for the eventual seizure of nearly 11 million acres of land from 250 sovereign tribal nations to establish land-grant universities across the United States, including many, many thousands of acres of land taken from Oregon tribes that established universities in states as far away as Maryland. Our university is located within the tribal homelands of the Marys River Band of the Kalapuya, who were forcibly removed to reservations in western Oregon. Today, living descendants of these people are part of the Confederated Tribes of Grand Ronde, Community of Oregon, and the Confederated Tribes of the Siletz Indians. We acknowledge the injustice and recognize our place as settlers on this land. We aspire to progress as an institution that seeks to better understand and address critical issues of equity and justice. The Gilfillan Lecture, a long-standing tradition in our college since the early 1980s, brings us together every year to celebrate outstanding science at OSU in honor of Doc Gilfillan, whose record of extraordinary science has positively impacted generations. I was very fortunate a few weeks ago at Drexel University to meet Paul Jensen. Paul is Doc Gilfillan's grandson and is provost at Drexel, so we had a nice time talking about Doc Gilfillan and his many memories. Paul, if you're joining us tonight, welcome. Doc Gilfillan served as Dean of the College of Science for nearly 25 years, from 1938 to 1962, and as acting OSU president from 1941 to 42. The Gilfillan Memorial Award honors Doc Gilfillan's life and recognizes his extensive service and contributions to Oregon State University and the field of pharmaceutical chemistry. His contributions are a lasting inspiration to us. I want to thank members of the Gilfillan family who've joined us today and their generous gift that enriches our college, Oregon State, and our community. By establishing the award, you are helping enhance our efforts to share scientific discoveries, cutting-edge research, and innovative teaching with OSU, our community, and the world, furthering our mission of science for all. And now, I'm very pleased and honored to introduce Dr. Michael Freitag, University Distinguished Professor of Biophysics and Biochemistry. I think I may have the honor of saying that for the first time. As our newest recipient as well of the F.A. Gilfillan Memorial Award for Distinguished Scholarship. So this award recognizes faculty in our college who demonstrate a long and exceptional scientific career, with nationally and globally recognized scholarly achievements. Michael is very well deserving of this prestigious award. He was named a fellow of the AAAS in 2019. He's described by colleagues as among, quote, the most respected scientists in the field of fungal biology and a worldwide leader and innovator. His work centers on fungal epigenetics and chromosome structure, and his lab is a magnet for researchers from around the world. Michael, whose field of research is molecular genetics, spans the areas of chromatin biology, gene silencing, and mutagenesis. He joined the Department of Biochemistry and Biophysics as an assistant professor in 2006 after working in the Eric Selker lab at the University of Oregon for a number of years. In his 16 years at OSU, he's developed a well-funded research program with continuous financial support totaling now more than $7 million, including current funding from the National Science Foundation, the National Institutes of Health, and other agencies. He's published well over 100 papers in journals such as Nature, Science, the Proceedings of the National Academy of Sciences, Nature Communications, and Genome Research. And he's received more than 13,000 citations for his work, which is a remarkable number. His talk, Our Lives Among the Peaks, Tales of ChIPs, Enrichment and Decay. Michael will outline the personal and scientific journeys that have brought him from Germany to Oregon, discuss the underappreciated benefits of fungi as a research tool, highlight how his foundational research paves the way for work in agriculture and biomedical research, and I hope finally settle the matter of whether it's fungi, 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 or fungi. Michael, congratulations. We're looking forward to your talk.
All right, thanks for that wonderful introduction, Roy. And it's pilz, actually, not fungi, not fungus, it's pilz. So hongo will work too, or hongo. So actually, before we started, Kari van Zee, one of my most favorite colleagues, gave me this nice, I love mushroom cups. There wasn't enough room for an S, apparently. So it's the singular. But I like mushroom or mushrooms, I like them all. Okay, so we talk about lives amongst the peaks. And I mean the peaks that you see here, Mount Hood, Mount Jefferson, and in the back the Three Sisters. Those we can see on a nice day where we live. It's a wonderful area. And I just came back from Germany just 2 days ago. And I always love coming back because I've been here for 35 years. Oregon is my home. It's the best place to live in the world, I think, after traveling for a long time in many ways. So what you'll see today is a scientific journey of 38 years, 16 of them at Oregon, and I have the opportunity to go overtime because there's going to be another talk in 2 weeks. It'll be a different talk about the distinguished professorship, and I'll advertise that as we go along. But I started when OSU's logo looked like this, with the Computational and Genome Biology Initiative, and I'm clearly nervous. Hold still. And I end here with Oregon State looking like this, and in between there are a lot of fungi. I'll switch over to fungi once in a while. Okay, so we talked about Dr. Gilfillan, Doc Gilfillan, quite a bit, and Roy already said a lot of things about him. He was a true Renaissance man. After receiving a bachelor's in pharmacy and then his degree at Yale in chemistry, he became a professor at OSU in 1926 or 27. Started teaching chemistry and pharmacy. He taught German and Russian, as seen there. He spoke fluently several languages, including Chinook and Chinese. This is just remarkable to me. I can just barely hold two in my head in the same spot, I think. He was the dean, as our dean just mentioned, but he also fostered the Junior Engineers and Scientists Summer Initiative, very similar to Summer Academy, I guess, for high school students. And he was a founder or a supporter of OMSI very early on. So a remarkable person, I think, and somebody we can only hope to emulate. So he was very active as the College of Science Dean. So he is inspecting a new electron microscope. He is inspecting a German Stein. He collected silver, and he is just about to discover that that thing is empty. So I hope it'll be different. He is in 1960s, just about the time when I was born. And after that, after he retired, he kept on teaching in the college and was at OSU for a remarkable 60 years altogether. So thanks again to the family, and we hope to continue his legacy here at Oregon State. So when you walk around Corvallis on a day when I have to take a car rather than my bike, you can actually run into the house that he first lived in. It's between Jackson and Van Buren, I think. It's the F.A. Gilfillan house. It says 1926 on it. I think that's the house. It says it. Okay, so how did I get where I'm standing now? There are 3 sections to this talk, and it might go into overtime, I can already warn you. So I studied forestry in Germany. So I went from trees to fungi, and then we're going from fungi to genes, and then from genes to chromatin or whole chromosomes. So for the students in the audience, both here in the room and also online, and I noticed there were like 10 or 20% students, so thanks for being here from the lab course and listening to this. You don't have to follow a linear path. You can wander, you can change your directions, and you'll still get to be a cool scientist if you want to. So where do I come from? I grew up in Germany, in the state of Lower Saxony, and the little thing you see here is the town sign. It's the town of Hamelin with a millstone and an old church that indicates it was founded in 1980. It's in central northern Germany. Hanover is where I was born. It's not in New England. It's this one, the real one. And my grandparents lived in Barsinghausen and in Lehrte, and I grew up mostly in this region. All of this is a diameter of from Hamelin to 35 miles or something like that. And my grandparents, at least 2 of them, really never left that area. And so my whole family was really not scientific in nature. I mean, my dad was, and we'll see him in a minute, was an engineer by trade in the end, but most of us were farmers or carpenters or mechanics. And most of the women stayed home to raise the family or work on the farm. And so Hamelin, the town where I lived in most of the time, is famous for the piper, the Pied Piper. And that's a cautionary tale about what happens when you don't pay your bills. Your children will be taken away. It's very rough. So we didn't get taken away. So here my grandparents, Harry right here, he was a chemical or an analytical chemist for a potassium mine, and Ernst was a mechanic. And here's my mom with her 3 sisters. My dad had 1 brother. I don't have a good picture of him. Here's a picture of me when I was about 2. I used to have hair, so it's hard to believe it was blonde. My mom, and here's my dad and my mom in just about 1955 or so when they first got married. And my dad was a builder and tinkerer and engineer. He had lots of hobbies, but I sort of inherited that spirit of always trying to mess around with things. If you ask people from my lab, they will tell you, we don't buy kits. We just try to do everything as much as possible on our own. Here am I at 6. I was not a good student. I was a terrible student in high school and all through my career. I just barely scraped by, but I made it into college, and I was the first one in our family to go to college, really. And here are my parents in 1991, my mom, Rosemary, and my dad, Heinz. And I inherited, or I actually learned, it wasn't inherited, I learned my love of reading from my mom more than my dad, and the tinkering from my dad. That's really all I want to say. Okay, so what did I do? After high school, I was part of that generation when it was cool to be green in Germany and be related to nature and the environment. And so der Wald had always a very romantic notation in Germany and in German poetry. And so studying forestry was sort of the obvious choice at the time, learning about ecosystems and maybe learning how to manage them responsibly. Sustainability is a thing that was sort of invented in Central Europe, in Switzerland and Germany. And so that was the thing to do already there. So I decided to go to Göttingen, and that's the Gänseliesel fountain that is usually scaled when somebody gets a PhD, and you see the flowers up there. So some lucky person just got a PhD and had to kiss the Gänseliesel. And the dream was that it would look like this. You would be in a forested area and learn about the forest. The reality looked more like that. So the forestry school is right up here. This is the place where you had to eat. This is microbiology. This was chemistry. All this stuff is new. This is a newer picture. Here's the Max Planck Institute for Biophysical Chemistry. That's where I would be now. Ha! That would be great. So, but okay, that was forestry. And after about 3 years or 4 years, I got sucked into a lab, and that was on fungi, because what people were trying to do at the time already was to use lignin or wood to make different products. For example, glues. Turns out when you put a glue that's made with fungi and lignin composites on the outside of a house, it's not waterproof. It doesn't work very well. So, but they wanted to learn how to use lignin in other ways. And so Alois Hüttermann here, he was running the institute. He was a biochemist himself, but he ran one section of the Botanical Institute, and he let me do a diploma bite, or an honors thesis, if you will, in his lab. And this was on decolorization of the dye polyblue 411 as a proxy for lignin, essentially. So here's the dye structure, and it's a polymer, of course. And so you were looking at absorbance at 482 and 592, and calculate the ratio between 592 and 482 to see that the dye was decomposed over time. And so if that worked well, then it would change from this pattern up high to lower patterns. And so 592 would go down. So this is a good decay. And Heterobasidion annoson, which is a common fungus on spruce in Germany and also exists here, wasn't so good at decaying this dye. So then over time, you take this ratio and you plot it over the days. And so I did this with about 15 different fungi in pure culture. And so that was my first venture into very simple biochemistry, essentially, in pure culture and looking into the medium, the growth medium. And so some of these fungi weren't very good, others were good. I was promised that would be a publication. This is the first time it's been shown to anybody. So doesn't always work out. So undergrads, watch out what you're getting promised. Okay, so after I was done, I really knew that I wasn't going to be a forester in the sense of being a district forester or a national forest director or anything like that, because there's too much law involved, too much business. So I went to Australia for a while. I traveled around, worked there. But then I got a message from Bob Kramer, who was then the department head in forest products. And he said, oh, you want to study wood decay? We have a couple of people who do wood decay. And so OSU truly had an open door. Here's the friendly Benny. I much prefer friendly Benny. Open door, it was true. This is my old passport picture. So I ended up going to Corvallis. I thought, well, maybe I'll do a master's here and then I move on. But I found very quickly that there were a lot of things I had really no idea about that were happening in biology. So here's Jeff Morrell. He was my master's thesis advisor. And I picked a pretty tough project. He let me pick my own project, but it was not trivial. And I learned from Jeff that writing really means editing, and that was in the sense of him changing things, and I changed to the things that he suggested, and then he changed it back to the things that I had previously written. So I learned that I don't want to do that. But Jeff overall got me on my way, and in one of my first classes, I met Everett Henson. And we did a reading and conference class that was on the biological species concept, and I thought, that must have been settled since, I don't know, 1930. What is he even talking about? And I learned that lots of things have to be re-examined every 10 years or every 15 years because things change. And there's a line I could quote from the Big Lebowski, but I won't. And so you have to go and look at old concepts again over and over. And then the last person who's flashing up here is Peter Bottomley, because I took his microbiological, microbiological ecology course. It was really interesting because I bought this textbook for $45 or something. I never used it because he taught everything from the primary literature, which is what I do now. And that's what we all should be doing. Textbooks are sort of crutches. They're the cliff notes of everything, and they're old when they come out. So primary literature is what we need to understand the forefront of science. So those were mentors during my master's degree at OSU. And then I met a lot of important people in Jeff's group. The first was my first office mate, Maureen, then Mitchoff, now Putman. She has not aged in all these years. This is a picture where she won an award. We got along great, and she introduced me to Camille, who's my wife, and without her, obviously I wouldn't be standing here. Let's go to that later. Jeff is inspecting a pole here. He's curing it by laying on his hands, I think, from wood decay. This is not a trivial task. So we're looking at decay fungi up here, and this is a trichoderma, a different group of fungi. And the idea of my thesis was to use the trichoderma group of fungi as a biocontrol agent to keep the decay fungi from spreading. That did not work at all. And so what we did in the end was to rescue the topic by co-culturing in liquid culture and then measuring enzymes. And I spare you a lot of graphs that were in the master's thesis. But it was a great learning experience about, again, doing biochemistry in situations that weren't perfect for biochemistry because that was not a biochemistry lab. But I met Camille, which was much more important than doing the work. So now we're going from fungi to genes. And if you paid attention, you saw the structure and the name Gold right here. So this is Mike Gold. And I wanted to work at the Oregon Graduate Institute of Science and Technology. And I always say more letters in the name than students. So it was a graduate school where there was mostly material sciences and computer sciences, but also a very active biological chemistry group. And they invited more and more geneticists because they wanted to learn how to do molecular biology. And Mike Gold was one of them. But when he saw me and looked at my portfolio dossier, he said, well, you're a biologist. I'm a biochemist. And so why don't you work with this guy who's coming from Stanford? And I came at the same time as this guy. But before I show Matt, I'm showing the typical lab attire in the lab. So we were in this outpost of the place, and it was a little trailer, and it was about 32 degrees in there all the time. You didn't need an incubator to grow fungi. And but honestly, I just came back from a soccer game at this time. But here's Matt wearing a typical lab attire. So he has this his counter for reactivity, his inevitable timer. These racks are from Beadle and Tatum days. They came from Stanford. And Matt, this actually flipped. Matt is a right-hander. He's looking at a tube of neurospora right there. So when I got to Matt's lab, I made a huge quantum leap in my understanding of biology because I had been an organismal biologist at best. I don't even know what sort of biologist I was. But now I did become a molecular biologist because I learned that you could take fragments of genes from all sorts of different organisms and put them together in a fungus and they would work. It's just crazy stuff, really. So to understand how much the field had moved forward without me knowing it all, that was amazing. And so working with Matt, I met incredible scientists like Bob Metzenberg, who is instrumental in figuring out phosphate metabolism regulation, the National Academy member, Roland Davis, who's working on the same protein that we were working on, and gene, who learned how regulation can be controlled by sequestering pathways in different organelles, and Charlie Janowski, who is my scientific grandfather, both from Matt's side, where Matt was a postdoc there, and from Eric's side, where Eric was a PhD student of Charlie's. And Charlie, of course, figured out that genes and mRNA are collinear, and if you had any questions about tryptophan, he could tell you what the answer was. So then the last person was Hirokatsu Inoue, a Japanese scientist who worked on DNA repair. And all of these were instrumental in helping me along to get my PhD in Matt's lab. So this picture is shown for two reasons. The first I have to set the stage. So most of you think of genes as encoding one mRNA, and then eventually the mRNA is translated into a protein. And that's true for most eukaryotic mRNAs. But many of them, and they're instrumental ones, they have the capacity to encode for two proteins or two peptides. And in some cases, that just regulates what happens to the downstream, the major protein. So if you have a small upstream open reading frame like this one right here that I studied for those 4 years, you can express this upstream open reading frame during translation, and that will inhibit the production of the main or the ARG2 protein that's instrumental for arginine biosynthesis. So a lot of these mRNAs that have two potential peptides encoded in them are important as growth factors or in other key pathways, and they're almost always the rate-limiting proteins in the end. And so that is functioning to control protein production very quickly and respond to environmental stresses in a quick and important way. So that was essentially my thesis work, and it centered on figuring out whether we could mutate any of these residues and then abolish regulation. And if you hit this D and change that, you think because it's so conserved, that probably would do something, and that's what the sequencing gel shows you. Yeah, they're sequencing gels. I ran those. So you read them from the bottom, and when you're in the dark room at about midnight and you see this one in the lanes that are the parent or the wild type, and you read up, and you get to the one that's the D, the GAT right here, you say, well, great, the control worked. And then you have the next lines over, the next lanes over, and you read up and you say, holy AAT, I have my mutation. It changes a D to an N, an aspartic acid to an asparagine. I can graduate. So that was not just that, but it was also the first time that I got this excitement that you get when you see something for the first time, and I'll come up a couple more times. So that's what keeps you going, I think, as a scientist. You make discoveries. There might be small things, just like this D to N thing, right? But on a personal level, they're important. The other very complicated looking picture is something that Gary Merrill probably recognizes. It's the polysome profile. It reads the other way around because I push my gradients out the other way from most people. But this, I don't want to explain all of this. I just want to show you this because there's a story in this, in that when you do an experiment like this, it takes 3 days. If you do not label all the tubes that you're carrying around in your arm, and somebody comes to a closed door and you can't see that person and all the tubes fall on the ground, you have to do the experiment again. So I would advise every student who's in the room to label all the tubes, because otherwise you'd be very sad. Okay, that's all I want to say about this. So the other thing I want to say about complete imposter syndrome is that I got a PhD from a school that no longer exists. So Oregon Graduate Institute of Science and Technology looked like this when I was there, and we were starting out in this trailer, and then we moved to the Cooley Science Center, probably named after Rick, I don't know. And then in, I don't know when, 1990-something, the OHSU took over OGI because OGI fell in financial straits, and then later on Microsoft bought it, and so all the buildings that were the old OGI are now bulldozed, and they don't exist any longer. So again, I really have to thank Matt for the times in this lab. It was an awesome time there, and I learned so much in so little time. It was incredible. So then we go from genes to chromatin, and this was a fortuitous thing because I went to what I thought was the last fungal genetics meeting I ever encountered, and I met Eric again, Eric Selker, who's from Eugene, still in Eugene, and he said, well, I need a postdoc. It's like, great, I don't have to move. And he did very interesting stuff at the time. So what you'll hear about now is stuff that has to do with epigenetics or DNA methylation and chromatin biology. But what I want to start out with were the things that really changed how we do science in very dramatic ways. So everything I told you up to this point was laborious and cumbersome because we're working on one gene at a time, one transcript at a time, and it was difficult to get to all the different things that we wanted to work on. But when you have a genome, to pick the genes that you think might be important, you do reverse genetics, you make a guess, and then you check out whether your guess is correct. Things can speed up. And I have 2 stories about that. So I was lucky to get together with Matt and Eric, and we decided to push some folks, together with many other folks like Jay Dunlap and Kathy Borkovich in the neurosper community, to have Bruce Byrne and James Galligan and Eric Lander, who's here as well, at the MIT Broad Institute to sequence neurospora. And this was the old school sequencing, difficult Sanger sequencing. But it only took, I think, 3 years from getting the go-ahead and finishing it. And a consortium of people, of course, do these things. Matt and I made the DNA for this, and I annotated a lot of the genes, as we'll we'll hear about maybe later. But the paper came out almost exactly 50 years after the announcement of the DNA structure, so that was fun. We didn't get the cover because of that, of course. There was the DNA structure to go on. The only companion paper at the time, often there are companion papers, was the one on the Neurospora methylome, as we would call it now. Well, Eric and I weren't so clever. We just called it methylated component. That didn't stick, so methylome is much more obvious, perhaps. But this was a collaboration that Eric started at a sabbatical time with Adrian Burt at the University of Edinburgh. So Eric isolated methylated DNA and then cloned the pieces, and we sequenced them in Eugene. And of course, you couldn't sequence very many at a time, so we only had about 200 of them. And when Brian Margolin actually looked to see what this DNA is, he always hit mitochondrial DNA, and we thought that's kind of crazy. Until we realized, well, you have to translate it and see what it is. And so we did, and it turns out all of these AT-rich regions that are shown right here that are methylated, because this band is high and this is low, that's the difference. This methylated, if it would be all the same down here, it would be unmethylated. But this methylated DNA is transposon DNA that's heavily mutated and AT-rich. And so methylated regions in Neurospora, as it turns out in plants and in us, in mammals, are transposon DNA is methylated and mutated. And so that was a key insight. And Camille actually helped me pick out names for these mutated transposons. So we came up with lollygag and listless instead of restless, and no-go instead of Tigger, and all this sort of fun names. When you have transposons, you're allowed to be funny. So that was good. But the genome itself was not really finished. In fact, it's quite the ill-tempered genome because there are so many AT-rich regions. And so it took until this year, until Andy Klocko and our lab, together with the JGI, finally got a telomere-to-telomere genome of Neurospora together. And it's not a large genome. It's only 43 megabases. So there were many other papers that followed that have to do with genomes. And so Roy mentioned that. I work on a lot of fungal genomes, and so they started out in journals like Nature. Then they got a little bit cheaper because there were more genomes, plus genetics and so forth. One with Linda Ciafetti here on Pyranophora. And then here was so it turns out, second de novo assembly of a eukaryote genome just by short read sequences, the Illumina sequencing that we all use now. The first one was the panda, the giant panda. I hate pandas. So and then the last one that we actively contributed to was Fusarium fujikuroi. That was a big collaboration with German labs. So the genomics and understanding methylated DNA was instrumental to get me started here at OSU. And the other part had to do with being a bit lucky and figuring out how to make GFP work in Neurospora. And Neurospora is a very actively researched organism as a fungus. And a lot of people wanted to do cell biology, but it was difficult until we could use the power of green fluorescent protein. And I ran into that in the hallways in Courtleigh Hall because Linda Ciafetti had actually gotten this to work for many plant pathogens. And I said, why can it work there and not for us? And so she kindly lent us the gene to make that happen. So here's also how Neurospora grows. It's a filamentous fungus. This is sped up about 10-fold. It's a fast fungus. If you look under the microscope, it grows through a cover slip image within about 10 minutes or so. What you see in green are all nuclei. And to a mammalian cell biologist, this is total anarchy because there are lots of nuclei in one compartment. They're not really cells. You see there septa developing here, the cross walls, but there's connection between compartment to compartment because those cross walls are open. And so the nuclei flow forward, that's anterograde transport, but they can also go backwards. There's a lot of stuff happening. But it gives you an idea of what the fungus grows like. It's also an awesome organism to look in the sexual cycle. So we do, with Neurospora, we do genetic crosses. It was the organism with which biochemical genetics was invented by Beadle and Tatum. And so here you can see one-to-one segregation. If a marker is closely linked to a centromere, you get inheritance according to normal segregation in Neurospora like this. If you see 2-4-2, you know there's a weird crossover occurring. Here you have some spores that are hyaline, so that the normal pigment isn't developed, and so you can look at synchronous cell division, very good tool. Here's one banana spore. 8 spores are just in one big compartment, and you can see how the nuclei that are over here that have the GFP, analogous to this, will actually make the GFP first, and then it slowly moves over into the rest of the banana spores. You can look at kinetics if you want to. So this was the first paper on this, and then there were many, many other papers on cell biology in Neurospora with microtubule dynamics, bulk flow, actin, more microtubules. Here's Thomas Liu. At the end, he was my first undergrad with Grant Farr when we started the lab. He got onto this paper. And then we worked with especially Nick Reed. He'll come up in a little bit on mitosis, and with Marichal Ricalme in Mexico on chitin synthesis and polarized growth and the exorcist complex for polarized growth. But all of this was helping me to get my own career going. What was going on in Eric's lab was the study of DNA methylation. And when I started there, we didn't really know all that much. We knew that there was repeat-induced point mutation. That's actually what makes these transposons so AT-rich. We'll get back to that in a minute. And then this leads to de novo DNA methylation. They observed that, but they didn't know how. And then there were a couple of unknown steps. There had to be a DNA methyltransferase, and that somehow needs to be maintained. So there should be some sort of cycle. So I never really wanted to work on repeat-induced point mutation when I got into Eric's lab, but I ended up working on it anyway. This is an interesting system that exists in fungi, but not as far as we know in other organisms. And it's there to prohibit transposons from doing damage. So here are 2 strains of a fungus, and when they are compatible, they can fuse. And now you have one cytoplasm, but 2 different nuclei, a heterokaryon, different nuclei. And these have different components. So let's say these are transposons that have proliferated. Now you have 2 or more of them, and something magical happens. We think it's actually pairing that's based on the DNA structure. And when that happens, an enzyme introduces CG to TA mutations. So they're always the same polarity. You never see anything else, usually C to Ts. Then you go through meiosis, and at the end, you have 4 meiotic products. And if the mutations, if you have a normal gene here instead of a transposon, are recessive, then 2 will be normal cells, and 2 will be mutated, and the genes will be heavily methylated. So that's what we knew about. Repeat-induced point mutation generates mutated DNA that's often methylated. And so the hypothesis then was that these CG to TA transitions could occur by methylation of the cytosine to methylated cytosine. And that could be deaminated to a T, and that would be fixed as a TA. So you have the CG to TA. That was the hypothesis. What enzyme could do that? Well, a methyltransferase could. And so we found one extraneous methyltransferase in the Neurospora genome, and I mutated that. Here's a DNA methyltransferase domain, and here's another domain that Ali will recognize and loves, the BA, brome-adjacent homology domain. And so the result was when we mutated RID, we did no longer see this effect, and none of the progeny were methylated or mutated. So in a homozygous RID cross, where both parents are deficient for this methyltransferase-like protein, this process is completely absent. So that was the first gene or protein that was shown to be absolutely essential for RIP, for this defense or genome defense mechanism. So keep that in mind. The second story has to do with formation of heterochromatin. And that's about a protein called HP1 that was first found in fly, Drosophila melanogaster. Looks very similar in structure to the Neurospora version. It has a chromodomain and a chromoshadow domain. This domain binds to methylated lysine amino acids, and especially in histones. This chromoshadow domain is an anchor to bind to other proteins and do its business. So in fly, there's no DNA methylation, but they had shown that this has to do with silencing. So I mutated this in Neurospora, and I found that it's essential for DNA methylation. So without this protein, there's no more DNA methylation at all. And together with Patrick Hickey and Nick Reed in Edinburgh, we actually showed that the HP1 protein occurs only in heterochromatin, and that's what this movie shows you. So the nuclei are being dragged along by microtubule spindles, which we can't see, and the one strong spot that you see in the front is essentially the centromeres all together, so the place where the microtubule spindles would attach during division. And the cool thing, and the second time that I had this aha experience as a scientist was when I observed this under the microscope. When the histone H3 methyltransferase was lacking, HP1 was no longer localized where it should be. And so that made sense in terms of a controlling pathway. And so the working model when I left Eric's lab was that RID was causing RIB, and that led somehow to de novo methylation that was requiring a histone methyltransferase that Hisashi Tamaru had worked on in the lab. HB1 would bind to the methylated lysine and attract the DNA methyltransferase, which Shinji Honda found out a little bit later. And then this was maintained in a way that involves a couple of complexes I don't have time to talk about. I also worked on what turned out to be a mutation of histone H3 itself that also resulted in absence of DNA methylation, and we excluded that RNAi or RNA interference was important for the process. And that probably helped me in finding another job because I think it attracted the attention of Jim Carrington when he was here as CGRB director, and so he was instrumental, I think, in getting me to come to Oregon State University. So the big questions we had when we started the lab in 2006 were really, how do genotypes yield specific phenotypes? So we think about, as biologists, we know that both nature and nurture are important for creating an organism. But how do we actually go from a genotype in an organism as simple as Neurospora or E. coli to a specific phenotype? This is not a trivial question to answer still, even though we have a lot of powerful genomics tools. We often don't even understand how cells alter expression, mRNA levels, based on extraneous signals or internal signals. So that's what I wanted to study. And then I was interested in expression networks and how they are organized, regulated, and maintained. And lastly, how do genomes evolve? So how does the Neurospora genome evolve? So over time, we had many different projects in the lab, and I can't talk about all of these. Some of them you might encounter in the next talk if you come. But today I'll just talk about two of these projects in some detail. And the first one has to do with transcription networks induced by light, and that has to do with the circadian clock. And the other one has to do with transcriptional memory or facultative heterochromatin that can be turned on and off at will, and that's really important for development and differentiation in us, but also in fungi and plants. So let's start with the Neurospora clock. So other than fly, Neurospora is probably the best genetic model for circadian clocks. Clocks always work the same way. You have a Zeitgeber, a time setter, that could be light, but it could be also very rich food, especially when you travel back from Germany to the US. You want to eat at the right time to make sure that you don't have much jet lag, and it worked for me. I'm not tired at all. So you have transcription factors that are activated with the Zeitgebers. In our case, in Neurospora, white color 1 and white color 2. In human, it's BMAL and clock, or in fly, cycle and clock. They form a complex, and we think in our lab really that white color 2 is the only one that really contacts DNA. They're activated by phosphorylation. They bind to a light regulatory element, and they actively cause expression of the frequency mRNA. There's also an antisense mRNA that regulates the amount of the frequency mRNA. So that's expressed, it's made into protein, and in human frequency would be purr or cry, TIMS maybe, fly has a period and timeless. And these proteins exist over time, then they get phosphorylated and then shuttled into the proteasome. And this all runs on a roughly 24-hour cycle, so 23.7 for Neurospora. There are also output pathways that are controlled by white collar complex, and these control metabolism, development, stress response. In fungi, unlike in cyanobacteria, where almost 100% of all genes are controlled by the clock, in fungi it's about 10 to 15%. It depends who you ask. So we thought this would be a great way to figure out how to use chromatin immunoprecipitation to figure out how transcription factors control expression. So all you have to really know about this is that this technique, ChIP, and this is where the chips come in now, allows the detection of nucleic acid-protein interactions, and we can identify sites. The cool thing is we don't have to worry about the proteins, which are pretty unstable. We use an antibody to drag down the chromatin, usually now just one nucleosome, as it's called, with the DNA attached. And then we get rid of all the protein, and we take the DNA as a proxy for where the protein bound. So this is one of the most commonly carried out techniques in the lab for the last 16 years. So you can do 3 different things with the DNA. You can do a conventional ChIP-PCR. You get very little information about this, and you have to know where to look. Then you can do a microarray, and very few people do that anymore, so I'm not going to even talk about it. And you can do ChIP sequencing, which is what we mostly do. You would take all the DNA you get, and you make it into a ChIP sequencing library, and you can sequence all of that. Millions and millions of reads are the result, and then you have to map them back to a reference genome if you have it, or you can construct your own genome if you want to try. So that gives you the most information, and it's unbiased. You don't have to know anything about the genome beforehand. So here's what one of those PCRs would look like. Here's input, and then you can check for enrichment of the protein, in this case white color 1 or white color 2, on a specific region. So here's albino 3, vivid, and frequency. And if you have really good enrichment, you see a lot of band here, but hardly anything in the controlled histone H4 band. And that's the input looks like this. This is great enrichment. So when Christina Smith did this, a postdoc in the lab and a good friend, she found that there were lots of binding sites, and I'm showing you one here in the original slide that was done with Chris Sullivan's software called CacheX. So we mapped all these reads to regions, and you get these peaks. They point down in this case, and they localize to previously found proximal light regulatory elements, or the antisense light regulatory elements. And when you look under these peaks, you find binding factors that you expect for these transcription factors. So this is the control. Control worked beautifully. I'm not going to drag you through 10 more figures. I'm just going to tell you the take-home. The take-home was that we had previously 4 targets that were known and well studied, and we found 300 more. Among them, 24 transcription factors that were controlling the next levels of gene expression that was conducted by light regulation or the clock. And people are still working on this. And we came from a light regulation network that had just these blue components to a much, much bigger network. And this was all really done by the ChIP sequencing approach, then verified by transcriptional regulation. So this is what approaches Hershey heaven. When he was asked, Alfred Hershey, what is his heaven? It's to have one experiment that works and do it over and over again. And so that's what we pretty much did for a while. So just a summary of the light work. So there are clock-controlled genes, there are blue light-inducible genes, but then there's this whole other group that we haven't even really worked on because not everybody seems to be interested in that. These are white-collar inducible genes, like FAR1 that Aaron Bredovic worked on, and then white-collar associated genes, where white-collar 2 seems to bind, but it doesn't do anything to transcription. But it might still do something important. It's just hard to study them at this point, so we haven't done that. But overall, this work led to a program project grant from the NIH, and my good friend Deb Bell-Peterson and Matt were part of it, and then also Jay Dunlap and Jennifer Loris, they're a couple, and they work on the clock at Dartmouth. So the last part of the talk, and I'm going to push the limit here a little bit in terms of time, will be on transcriptional memory and K27 methylation of histone H3. And for that, I have to talk a little bit about chromosome territories. So when you think of chromosomes, they're often drawn like these spaghetti things. So these would be 2 chromatids or chromosomes that are about to undergo mitosis. And there are spindles attached to these outer kinetochores. Those are the protein complexes that bind to centromeric regions that are DNA. And all of this is constitutively silent. There are no genes expressed in most organisms in these regions. There are some exceptions, but mostly there are no expressed genes. There's some subtelomeric regions of heterochromatin, that's different chromatin, that's always silent. And there's subtelomeric rDNA that's also usually silent, but some repeats of the ribosomal DNA are expressed. Then there's euchromatin, normal for euchromatin, that's expressed under most conditions. So all in green here are the genic regions where the genes are localized, and they're expressed under most conditions. And then we have this interesting facultative heterochromatin that I want to spend some time on now. So that's in orange. This is the silent chromatin that needs to be activated under certain internal or under certain conditions that are either controlled by internal or external stimuli. So chromosomes obviously don't look like this, and so for a while we worked on high-C, which is a method to essentially model what chromosomes look like. And John Galaska, who was a postdoc in the lab, collaborated with Andy Klocko and Eric Selko's lab to make this happen in Neuraspora. So if you had an expected contact frequency of a chromosome that looked like the one that I showed you before, just a linear piece of DNA, you would see alignment like this. Everything which is its nearest neighbor would have contacts, but there would be no contacts, say, from here to here. That would show up right here. So this is the expected contact frequency if you expect no contacts in a chromosome. But what you get, if you look at a whole genome, is this sort of pattern. So, for example, here are centromeres that interact with each other, like I showed you in the movie, as the nuclei are being dragged along. And so all chromosomes share the centromere as one interaction point in this graph. And when you model the individual chromosomes, you can see the centromeres are extruded a little bit from the rest of the chromosome arms. And the telomeres are near each other as well. And so you sometimes see these little spots for the telomeres as well. So you can learn a lot by essentially just doing sequencing under the right conditions. I don't want to go into more detail. But John developed this technique, and it also helps to assemble genomes. And that's what I showed you on the previous slide that had this pattern. So how are the chromosomes controlled in terms of expression? What makes them active or silent? So people think of a histone code that aids to help with that. That's a good hypothesis. We don't really know that such a thing as a true code exists. But when we talk about histone codes in the context of histone H3, so these are the proteins that make up what's called a nucleosome, and then DNA is wrapped around it several, 1.7 times or so. So histone H3 occurs as a pair together with H4, H2A, H2B, and it's modified on its amino terminal end. And here they're just lysines shown that are modified. And they can be either methylated or acetylated, and I just show methylation here. If lysine 4 is methylated, then you have usually transcriptional activation or genes are expressed. If you have lysine 9 methylated, HP1, our old friend, can bind. If it doesn't exist here, then HP1 cannot bind. Remember the microscope slide where it was just all over the nucleus? So here you have binding of HP1 to lysine 9 methylated, and that's catalyzed by KMT1 or lysine methyltransferase or DIM5. And if you have lysine 27 methylated by lysine methyltransferase 6 or SET7, you get transcriptional memory of facultative heterochromatin. The other marks we'll talk about on May 20th. So ChIP sequencing in Fusarium turned out to be very interesting. So here's now the profile of a ChIP experiment, and what we do is take the DNA reads and we stack them up on top of each other in form of a histogram. And so you can see the distribution of the active histone mark across the chromosome. And when you look at H3K9 methylation, you see there's very little of it. There's a little bit at the centromere and close to the chromosome ends. But H3K27 was a real surprise, and that got us started onto a new tangent about 10 years ago. And there was so much of this histone mark, about 4 times more than in mammals or in plants. And so the question was then, is gene silencing controlled by this K27 methylation like it is in flies and in humans? And which genes are enriched in these regions? Are they important? What do they do? So we studied this by comparing the Fusarium graminearum case to that of 2 other species. And what's shown in different colors here are the chromosomes of these other 2 species. They have 12 or 14 chromosomes. The species we study, Fusarium graminearum, has only 4. So if you look at these patterns, you think that would be the end of a chromosome. This would be the end of a chromosome. When we mapped the K27 methylation onto this pattern, we saw that this epigenetic mark is localized in the subtelomeric regions of the ancient chromosomes. So K27 methylation as an epigenetic mark existed for millions of years in these separate species, and it's maintained in the same position. And it's in regions that are essentially also a little bit new. There are new genes found in different species in those regions. So the other discovery we made by looking at other fungi like Fusarium fujikuroi and then also Mareike Möller's favorite organism, Zymoseptoria triticea, was that accessory chromosomes that have a lot of usually metabolic gene clusters and other interesting genes are completely covered by K27 methylation. So they are sort of certain packages of extra capabilities in chromosome form that strains can sometimes trade and use for different purposes. So the last question that we wanted to address here was to figure out, is KMT6, the lysine methyltransferase, important for gene regulation? So we deleted this gene, shown right here. There's no signal. And the K27 methylation goes down, and it's basically background. And when you put the wild-type copy back in, it comes back. So that was actually interesting for understanding control. And what happens is that overall expression in these regions goes up in the mutant versus the wild type. So you get more expression of most of the genes that were covered by K27 methylation. And it was 25% of the genome, a staggering number. And it turned what looked like a husky fungus into a beaver fungus. It became orange and had very strange phenotypes, which we still don't really understand. Is this important? Is this just something that occurs in fungi? It's not. It was first found in flies. And polycomb repressive complex is the complex that's made up of KMT6, EED, and SUS12. And polycomb refers to the sex combs and the fly leg. But it's important for development because what's growing out of the fly head here are not antennae, but legs. So if you have a messed up system, PRC2, you get transformations that affect the outcome of development. And so when we deleted any of the components of PRC2, we don't see K27 methylation anymore. If we change the lysine to an arginine or a glutamine and the histone H3, we see something that looks like KMT6. And so we can also look at this by cytology to bring you back to microscopy. So we can see the heterochromatin on the inside of the nuclear membrane. This is a marker for the nucleus, for the nuclear membrane. So that's where the silent chromatin exists. And when you look at this in a mutant, you see there's no signal. So all of the components of the complex have to be around to make it work. So here's the, here's my plug for the next lecture. So come and see more about epigenetics. Epigenetics, I know it when I see it. So hopefully that'll be interesting. And the summary on fusarium heterochromatin is it controls development and differentiation. It serves as a model to understand mechanisms that are at work in plants and mammals that are difficult to address still in plants and mammals because things are so much more complicated than in the fungi. We found that it reversibly silences secondary metabolite clusters, and we worked with Sandra Luskin on this a little bit. It affects cellulolytic enzymes and some unusual metabolic pathways, and it also represses pathogenicity factors in the fungus. And so I got this quote actually not directly from Dr. Gilfillan. I got it from Janet Tate's talk, and I hope it's correct, Janet. So he said, if it's scientifically interesting, we will find a use for it eventually. That stuck with me. I think it's a great quote because we do exploratory science. I told you a couple of examples of hypothesis-driven science, but I love to explore and just find new stuff. I think there will be an important contribution by the fungi, 3 of them shown here, on understanding how this complex works in human and in Arabidopsis and in plants that are crop plants as well. And the last thing I really want to say before I get to the acknowledgements is that a lot of the work we've done, this will come up in the other lecture on the centromere, is really excellently situated for working with undergrads in labs. And some of you are here, so you're working on a project this year that has to do with interesting chromatin genes. Your colleagues in 2015, when I was on sabbatical, worked on centromere and kinetochore proteins, and Steve Friedman, who was a PhD student in the lab, and Kari van Zee led it, and Corinne Fargo was another teaching assistant. And here's another quote from Gilfillan, educate for tomorrow. That's what we really do here, right? So a lot of our students and trainees go on to work with other collaborators, and here's some of them across the world I have worked with over time. They were part of some of the studies that I showed you. Only Antarctica is missing, so I have to find somebody down there at the station. But we educate for tomorrow, and a lot of the trainees that I had in the lab went on to great universities and became either doctors or PhDs and professors in some cases. So here's the first lab, Thomas Liu. I mentioned him already. He got a paper. Grant Farr did not. Both of them are doctors, physicians. So is Paul Donegan. Christina is working at Cascades right now. Linnell left the lab a little while ago, but she's working with Laurent Deluc. She can't leave the building, apparently. This was a crew that stuck around for a long time. There's Kyle Pomraning, Pallavi Fatale, and Aaron Bredovec, the first 3 grad students who finished their PhDs. Kyle was amazingly prolific. I didn't really talk about his work today. Joe Whalen was the first student who graduated from the lab with a master's, and Greg Eckberg is also a doctor now. Christina was a postdoc together with me in Eric Selko's lab, and she became instrumental for building the lab from 2008 to 2010. Here's a more recent picture. That was the second, if you will, edition of the lab that was really important, with Fong and Sholan working on a screen to find new mutants in K27 methylation. Steve, as a new grad student then. Corinne also working on the K27 stuff. John worked on the high C. Then we had trainees from the Netherlands who worked in Germany, Klaas Rotanus. Talented undergrads, all from our department. Zach, an amazing student who's now at UCLA as a med student. Brian is a trainee at Oxford, almost done with his PhD in Ben Davis's lab. Sholan just defended her PhD at OHSU. Brad is working for a company in the Bay Area. So they all landed in good places, I think, over time. A couple more images of folks who came from overseas. Lena Stute, who is now in Vienna. Rodrigo Gonçalves, he came twice, and here's a party where we had a lab reunion on the dance floor at a SOMA meeting. Jorka just visited in Hamburg, and he was a farewell party for Brian, who left for Oxford. A retweet image. And then the most recent version of the lab, version 3, where Mareike with a German fellowship and John with the USDA fellowship joined as postdocs, and they've been here for 3 years. Here was the graduation party in 2020, where some of us had to be sitting very far apart, and we had pizza. So it was a little bit sad, but we had still fun. And pretty much at the end, and only remains to thank one person sitting right here for helping me making it all possible, and that's Camille. So I've been here for 35 years. I'm very happy in Oregon, as I told you, and I'm definitely powered by Orange. Neurospora. Thanks. So I guess I'm supposed to do the questions from here, so ask some. It's fungi, actually. Truly, fungi, not fungi. Often not. David, all right.
What is the difference between the WC2 REs and the LREs, since they're both bound by WC2?
So the light regulatory elements would be, yeah, that's a good question. So they would actually be determined by light regulation themselves. And the other one we called white collar binding sites because it didn't seem to have anything to do with light. And when you do these ChIP experiments, sometimes you just have binding of transcription factors to specific regions that doesn't mean anything. So Barbara Wold, who first pioneered that in animals, she gave a talk a long time ago here, and she pointed out, 20% of our binding sites don't seem to matter at all under the conditions where we're looking. So it's hard. So the things that weren't called LREs, we excluded in terms of having anything to do with light or the clock. Say something from Zoom.
Yeah, we've got Brett Pierce. He's got a question.
Oh, Brett.
How is PRC2 recruited in Fusarium in the absence of PRC1?
Oh, that's fun. Very, very fun. Ellie, do you want to answer that? So PRC1, polycomb repressive complex 1, this was not planted, by the way, doesn't exist in fungi, but it's important in flies and in mammals. So the question is, how does this complex PRC2 know where to go? And Ali is writing her PhD thesis on part of that. We found a protein called defective in silencing 2 that we think binds to SUS12, one of the components, and then helps to drag it to the right spots. But that's probably not the only protein that does it. There might be actually some transcription factors similar to the white collar transcription factors doing that. So it's something we've been working on for a long time. And we're still not that much closer to a real answer because the genetic selection that we carried out gave us over and over again just a couple of interesting genes and proteins to work with, but not a general answer on how the targeting is actually achieved. So, okay. Anybody want to talk about ChIP, enrichment, and decay? It has nothing to do with Dostoevsky or the gambler or anything. So okay. Andy.
You quoted Gilfillan saying, find a good experiment and keep on doing it, or was that not Gilfillan?
No, that's Hershey, Alfred Hershey. After the Hershey-Chase experiment, yeah, he basically was asked what his definition of heaven is and said, find an experiment that works and do it over and over again.
So you were saying that ChIP was that kind of an experiment.
Yeah,
For you,
that's true. So, I mean, I've had 3 legs to stand on. One was fungal genomics, or genomes in general, and annotating them and learning about specific pathways. The other one was cell biology with green fluorescent protein and red fluorescent protein, and helping folks at other universities to answer their questions by helping them with genetics and using Rossborough and other organisms to do so. And the other one was to look at chromatin factors by using chromatin immunoprecipitation to figure out where proteins actually bind to DNA and then making a good story out of it, right? So, Yaga.
Another quote by Valerian Dolia was, never repeat successful experiment. It
well.
will not work second time.
Well, so that is actually that is the story of this too, because we repeat it over and over again until we're really sure that what we're thinking is correct. And so, no, you should, undergrads and grad students, you should always repeat your experiments many, many, many times before you publish anything. You don't want to retract anything ever. Okay. Let's see, here's another one.
Another question from Zoom. How important is RIP in the rest of the tree of life?
Oh, RIP. So RIP is this repeat-induced point mutation again. And so Rossborough is super active in this process. So it really keeps this genome clean of transposons. There are only 2 known transposons, whereas our genome is full of them all the time. And so RIP, as far as we know, or anything like it, does not occur in mammals or even in animals in general. And it's also not been found in plants, but people haven't really looked very hard. So sometimes you find surprises. It doesn't exist in bacteria, so it seems to be limited to a specific group of fungi, ascomycetes, and there's something similar in basidiomycetes, but there's no mutation, but just methylation. So Eric Selke was always saying RIP is an epigenetic phenomenon because methylation is really important. But I figure if there's a mutation, then it's a genetic phenomenon, right? A mutation occurs, genome changes. So, but it exists, but largely in the fungi. But there are lots of organisms. I took the shrub of life out of the talk because it's already too long. But we have to consider that animals and plants are a very, very small branch, and that in many cases we haven't even looked at all the bacteria, all the protists that are out there. We have no idea what's out in the world, and we're killing it at a great pace. So we should consider that. All right, thanks.
So let's congratulate Michael again. Good job. For the students here, let me say, and I don't know if this will resonate with my colleagues who are full-fledged PhD scientists, but I'm not a biochemist. I'm a long ways from biochemistry, but 2 things about this. One is the beauty of it. It's just it's wonderful to see the beauty of science. I think there are so many excellent examples there. That the philosophy of it is wonderful. The beauty of the discoveries is wonderful, and the connections between so many different fields and subfields is wonderful as well. The second is that, and this is really for the students, is My guess is, and this will be true for many scientists here as well, who are PhDs and who have many years of experience, and probably even a few biochemists around, you won't have understood everything today. And that, I want to tell you, is completely normal. That is completely normal. It's what, in fact, for those of you who are more junior in your careers, maybe you're an undergraduate or graduate student, one of the most important things about science is learning to be comfortable with all you don't know. And in fact, Michael talked about that right at the end. You say there's just a whole bunch of stuff we don't know out there. And that is part of the secret of being a scientist, is learning to be comfortable with all this that you don't know. And in fact, you don't know most of it. So thanks very much, Michael. Enjoy the evening, everyone. Thanks to everyone online for joining us for this wonderful event. And we'll see you all in 2 weeks.